Hello, everyone. Thanks so much for joining us for the LAMA2 research breakout session. We have a really great panel for you today. Before we get started, I need to read a disclaimer statement. The information and opinions shared by our panelists will be generalized statements and are not intended to be interpreted as a promise for future treatment outcomes or a specific timeline for such outcomes. While we are all eager for advances, it is impossible for anyone to predict when a treatment may become available or anticipate roadblocks. Please be advised the session is being recorded and will be shared on our public YouTube channel after the conference portal closes on August 31st. You will also be able to watch this recording in a couple of days within the conference portal. Okay, so first we'll get started with introductions. And Dr. Bönnemann, would you like to introduce yourself?
I am Carsten Bönnemann, a pediatric neurologist and neurogeneticist at the National Institutes of Health, NINDS, and my research is in developing outcome measures and clinical therapies for early onset neuromuscular disorders, congenital mostly.
Great. Professor Muntoni.
Good afternoon. I'm a pediatric neurologist and I work at the Institute of Child Health at University College London and Great Ormond Street Hospital, and I've been involved with many of you for many years now.
Thank you. Professor Rüegg.
My name is Markus Rüegg. I'm a professor of neurobiology at the Biocenter of the University of Basel, and I'm working in preclinical research trying to develop therapies for LAMA2 deficiency.
Great. Dr. Foley.
Hello, I'm Reghan Foley. I'm a pediatric neurologist. I had the honor of training with both Dr. Bönnemann and Professor Muntoni, and my focus is clinical research for congenital onset muscle diseases.
And Professor Kemaladewi.
Hi, everyone. My name is Dwi Kemaladewi. I'm an assistant professor at the University of Pittsburgh. I started working on LAMA2 CMD since my time in Toronto with Dr. Veronica, as I got introduced to this wonderful community, and now I was able to continue on doing this in my own lab in Pittsburgh. I will be talking about development of therapeutic genome editing in LAMA2 CMD.
Great. And last but not least, Professor Pini.
Hi, I'm working with Francesco Muntoni in London, and the aim of our project is to identify modifier genes to ameliorate the disease severity in LAMA2.
Great. Dr. Bönnemann, I believe you have some slides to share with us.
Yeah, I just want to, I have just a slide with a few bullet points that allow me to link what we're talking about now to the morning session that I introduced some of the concepts, just so that we talk all about the same thing. So LAMA2 is the gene, and it makes the protein laminin alpha 2. Laminin alpha 2 is a very big protein, so LAMA2 has a very long coding sequence, meaning it's too big, too long to fit in regular AAV gene therapy vectors. Merosin is laminin 211, so it's a trimer, 3 proteins, of which laminin alpha 2 is the longest, but it also contains beta 1 and gamma 1, and then therefore it's called 211. Laminin 211 is the most important physical connection between the muscle fiber and its direct covering called the basement membrane. And breaking that link has a number of important consequences for the muscle fiber that makes it unhealthy and prone to disease. This link is all important, but could be provided by other molecules such as laminin 111 and other nifty tricks, as you will see today in the presentation. There is, as opposed to some of the other diseases, there is one mutational mechanism where it's recessive loss of function, either complete or partial, in this one gene, LAMA2. Mutations in both gene copies are necessary to cause disease and need to be known, but it's sometimes difficult to find both. And that's also an important point when we talk about therapies. And this, this is just what I just said. This is the alpha 2 chain here in the middle, and it's part of merosin or laminin 211, 211, beta 1, gamma 1. And this is the link here to the cell surface that is so all important. When it's gone, we have the laminin 211 deficient congenital muscular dystrophy. And what ends up being in the basement membrane is a different laminin, laminin 411, that does not provide the link to the basement membrane. So it's sitting there, but it's dysfunctional. And lastly, what I said, if you replace alpha-2 with alpha-1, you get laminin 1-1-1, and that is able to restore a linkage to the matrix. So these are just some of the fundamentals to prepare the session for today.
Great.
With that,
Professor Rüegg.
Yeah.
Great, we can see your slides.
Okay. Yeah, I will talk about linker-based gene therapy for LAMA2 muscular dystrophy. And just before I start, I would just to briefly show, because you're not familiar with this, we are now currently working in this old building of the Biozentrum here in this floor, and we're going to move in summer to this building, which makes us very exciting, and I'm sure we will continue to work on this problem. What I would like to do today is to show you a bit of an introduction and then go on and show you 3 slides on the current research, the progress we made on these questions, gene therapy, and using the linkers. Again, as Carsten has alluded to you, laminin 211 is a big molecule that is mainly expressed in the extracellular matrix surrounding the muscle fibers. As you can see here, this is laminin 211 surrounding all of the muscle fibers. But there are also other isoforms like this laminin 411 that instead of alpha-2 has an alpha-4 chain. And in a normal situation, it's expressed in blood vessels as seen here. But in the context of laminin alpha-2 deficiency, this protein now is also found in the basement membrane surrounding the muscle fibers, as for example, you can see here in a cross-section of a muscle biopsy of a human patient. However, as also Carsten has alluded to you, laminin 411 cannot compensate for the loss of laminin 211. And we have therefore devised small linkers that could eventually serve as linkers to hook up laminin 411 now again to the muscle. And we used agrin, which is a large molecule. We made it very small. We made this mini agrin with the idea to link laminin 411 now to the muscle sarcolemma. And we generated transgenic mice that express this mini agrin just in muscle. And we were able to show that this mini-agrin expressing laminin alpha-2 deficient mice, they, instead of dying at the age of about 10 weeks, they now survive for more than 40 weeks. In the second approach, together in collaboration with Peter Yurchenko from Rutgers University, we also tried to replace this function here, what also Carsten alluded to, that laminin 211 can actually form a polymer with other laminin 211 proteins. This again, laminin 411 cannot do. And Peter then devised another protein, a linker, which consists of parts of another protein called nitrogen and parts of laminin alpha-1, this amino terminal end. He generated a linker he terms alpha-LNND, and this then should allow also polymerization of laminin 411. We therefore expressed both linkers again in skeletal muscle in these laminin alpha-2 deficient mice. And again, as you can see, we get really a strong improvement also in survival. These mice now live for more than 80 weeks. Some mice actually live longer than their wild type littermates. Now in these experiments, which are all published, we have expressed the linkers just in skeletal muscle. And of course, laminin 211 is not only expressed in skeletal muscle, but also in other tissues. That's why we have tried to approach the question whether these linkers could eventually also help laminin 411 to become laminin 211-like in other tissues. And we have used for this purpose mice that express now these linkers ubiquitously. And here you actually see a movie of 14-week-old mice. This here is a mouse that has this laminin alpha-2 deficiency. That's a severe form of a laminin alpha-2 deficiency. And here you see a wild type control. This is this mouse, and this here is now a mouse that expresses these linkers not only in skeletal muscle, but also in other tissues. And you can see that it's actually quite difficult. They are a bit smaller, but they look very much like wild types, showing that these linkers in other tissues, in particular in the peripheral nervous system, are capable of replacing the function of laminin 211 in these deficient mice. The second approach, of course, if you think of clinical trials, you have to bring these linkers not transgenically, but have to bring them via vectors like AAV vectors. And this is what this shows here. In this particular experiment, we injected AAVs that express either the alpha LNND or miniagrin together into the facial vein of a P1 mouse. And then we look how they develop. And here you see a movie now of 6-week-old mice. Again, here, a mouse that is deficient of laminin alpha 1. Here, this one is the treated one, if I'm not wrong. And this one is a wild type littermate control. No, it's actually, this is the treated one. This is the wild type. And you can, it's also, again, a striking phenomenon that you improve here. The mouse is much improved. In terms of weight gain, also these linker expressing mice are heavier, significantly heavier than the laminin alpha 2 deficient mice. And we are now currently looking how long these mice survive. If we also look at more quantitative measures like fiber size distribution, you can see here that these laminin alpha 2 deficient mice that express these linkers, their muscles is improved. The size of the muscle fibers is improved. Inflammation, which is a sign of the disease, is way down in also these mice that express these AAVs expressing both linkers. And with this, I would like to finish and just quickly acknowledge mainly Judith Reinhardt, who's a senior research associate in my lab, who has done all of these experiments, and of course the collaboration with Peter Yurchenko, the funding by CureCMD to generate these transgenic mice, and currently we are funded by the Swiss government for developing this into a gene therapy. And with this, I'm done and would like to thank you for your attention.
Professor Rüegg, can I ask you a question? What would be the next steps then to translate this work from the mouse to the human?
Yeah, now we need, of course, now we have basically, I can stop the share. We have now shown that basically AAV can work. We have to maybe work a bit more on the details. And also, of course, now we have to develop ways to test for toxicology of if you express it, for example, in other tissues. But I think we are now in the way to try to drive this to eventually clinical trial. We need, of course, a pharmaceutical company to help us with this.
Great. Does anybody else have any thing to add to Professor Rüegg's presentation before we move on? Nope, we're good. All right. Dr. Kemaladewi, you're up.
Can you see my slide now? Yes, I hope you can see my slide. So
Yes.
I didn't have a cover, just
You
like
have to go into Dwi, you have to go into presentation mode.
Am I not in presentation mode right now?
The one to the right of it.
If you see presentation mode, you probably have to switch monitors.
Where is that?
Up in the top of the PowerPoint window, it says swap displays.
Oh, goodness. We tried this earlier. Can you see my screen at all?
Yeah, we can see your screen. Maybe just reduce the size of your side margin and that would be okay.
Let me try again.
Perfect.
Perfect.
Okay, cool. All right, so I didn't have the cover as what Marcus had. Otherwise, I can show you where the beautiful Pittsburgh is. So my lab here is quite new. We started in 2019, but then 2020 was canceled, so I'm still pretty much my first year. So the lab works on development of therapeutic genome editing for LAMA2 CMD with a focus on targeting LAMA1. As you already heard, that LAMA1 is the little sister of LAMA2. And what do I mean by that is that they are quite identical, and multiple works from the wonderful researchers in the field have shown that they can actually compensate for the lack of LAMA2. However, LAMA1 is not expressed at the same time as LAMA2, so it's only expressed during embryonic development. And what do we mean by that is that if DNA is actually a recipe book, our body actually has a recipe to make LAMA1, but it's actually too small, so we cannot see it. And if we cannot see it, we cannot use it. So what we wanted to do is to upregulate body's own LAMA1 using what's so-called CRISPR activation technology to compensate for the lack of LAMA2. So how does this technology work exactly? In a simplified way, you need to have 3 things, or 4 things actually, so 3 to 4. So the first one is called dCas9, that's the purple blob over here, that can be brought into a specific target in the genome, for example, mouse LAMA1, using this golden arrows called guide RNA. So there are 2 components already. And then we can also attach what's so-called transcriptional activators, which is kind of like the magnifying glass in the previous cartoon. So if you have all of this together in the body, then you can boost the transcription, a process called transcription of that gene of interest. And how do we deliver that to the body? That's the 4th component that you need, which is the viral vector. So we use viral vector AAV9, the same as what Markus used as well. And by now you know that AAV9 is a small virus with a small capacity. So to package all these components, we need to have 2 viruses. The first one carries the purple and the pink components, and then the other one carries all the guide RNAs. We then inject it into the LAMA2 deficient mouse. We do this in parallel or subsequently, almost subsequently, not only to the mouse, but also to the human DNA, because mouse and human DNAs are different. But to do that, we actually can still use the same technology. So the purple blob and the pink blobs are still going to be the same. We just use different guide RNAs. So now this is green, and that targets a human LAMA1. We tested this not in the mouse, but in the skin cells that were donated by you and or the community. I mean, the community, either directly to my previous lab in Toronto or to Coriel or to CMDIR. So thank you so much for the contribution. I can tell you that the cells move with me, so they are now growing in the incubator here in Pittsburgh. So yeah, we do that in parallel. And when we first did that, the question was just can we actually do it? Or is it something like a sci-fi? So we really the aim was really just, can we do it? And without going into too much detail, this is also published. Here in the untreated muscle, you don't see any LAMA1 in red, but here in the treated muscle, you start to see LAMA1. So this muscle does not have LAMA2, but now it has LAMA1. And we also look at the nerves. So in the untreated nerves, you do not see LAMA1, but in the treated nerves, you now have LAMA1. And then in the cells over here, you see that in the untreated cells, you don't have LAMA1 over here, but LAMA1 starts popping up in the cells that were treated with the CRISPR-Cas9 components. So can we do it? Yes, we can do it, but it's not perfect. Just like many other technologies, just not perfect yet. And what do I mean by that? Without going into too much technical details, the most imperfection that I think we need to improve is the use of 2 viruses, as I outlined earlier, because that directly correlates to the high cost and the potential toxicity. So moving forward, this is what we wanted to do. Now we can ask longer question: can we actually do it better, safer, and cheaper? So my lab now works on what we call the CRISPR activation 2.0. That should be stronger but smaller. So it involves a lot of molecular design. We can talk about it during Q&A if you are interested in. But the goal is to fit all these components, the blobs and the arrows, so that it can fit just into one single virus. We are trying to do this in a kind of like a semi-holistic approach. So looking at the muscle, the nerves, hopefully can target the brain as well. And then we also measure the breathing of this untreated and treated mice. With the cells, we are trying to look at mitochondrial properties and some other cellular properties that I won't have time to talk about. But we also want to, of course, not forget about the potential toxicity. So all the above, so all the internal organs in terms of pathology, as well as the off targets, which is pretty much how it affects other genes or other tissues in the bodies. The goal is hopefully to bring down the cost and to bring down the potential toxicity. All right, is that 3 minutes? So this is the team who are doing this under the mask, not behind the scene. So I demasked these two because these two are young students are those who actually drive this project. And I also would like to thank the Cure CMD for the support since the first time I work on this condition until now in my own lab. It's my first grant that I get, and then my first grant that I get also as an independent investigator. Thank you so much for the support.
I can't tell you how important it is to have both veteran researchers and young researchers in this field. It's an amazing collaboration. And so it really is driving this field forward. So we appreciate you doing.
Thank you.
Great. So we're going to jump into some questions now. I think Dr. Foley, we can direct this one to you. Do you know the status of the Omegapill trial?
Did you, Rachel, did you want to give Veronica a chance to present too?
She indicated she had no slides. Is that correct, Veronica? No.
Oh, okay.
Yeah, I don't have slides.
Right.
If you want, I can present the project in a few words.
Sure, go for it.
So basically, we started with the hypothesis that there are some modifier genes that can modulate the severity of LAMA2 disease manifestation. And this came from the observation that generally patients that don't have laminin alpha 2 at all in muscle, they have a quite homogeneous presentation of the disease. And generally, this results in the unachievable ability to ambulate independently. While patients that have traces of laminin alpha 2 in their body, they have a more heterogeneous spectrum, and you can either have patients as severe as those where laminin alpha 2 is absent, or patients that are milder. But we identified one particular case that is the only patient known so far, which is one of two affected siblings with the same genomic mutation that results in the complete loss of laminin alpha 2 in muscle. But this patient, despite the total loss of laminin alpha 2, is still able to walk at the age of 31, and he presents a very mild disease manifestation. And that's why we suspect that there are some genetic modifiers that are actually ameliorating the disease in this particular case. So what we did is to analyze the genes expressed in the muscle of this patient and compare it to genes expressed in the muscle of other patients without laminin alpha 2 in their muscle. And now we are at the analysis stage, and we're trying to identify genes that might be important for some muscle functionalities. And at the moment, we identified a few potential genes that might be interesting in ameliorating some aspects of the disease, like, for example, metabolism of carbohydrates and lipids. But these are not confirmed yet because we need to have further studies, and we're also analyzing the DNA of both siblings to see if we can correlate the gene expression level with particular DNA changes that are found only in this atypical patient. And then once we will identify a few genes that might be beneficial for ameliorating the disorder, we will test them in animal models. And in particular, we will test them in the dystrophic zebrafish. And we will try to analyze them either singularly or in combination and see if one of these genes or multiple combination of these genes together might result in the rescue or partial rescue of the disease in the fish. And this, this is working, it might help to develop new therapeutic approaches for the disease.
Great, thank you. And there is a poster in the posters and resources tab from.
Yeah.
Yep. So you can check that out and learn more about Dr. Pini's work.
Thank you.
Great. Okay, on to questions, shall we? Great. Dr. Foley, can you give us a status on Omegapill?
Sure. So we had been a single site here at the National Institutes of Health for a phase 1 safety and pharmacokinetic study of the oral compound Omegapill, an anti-apoptotic or anti-cell death compound tested between patients with either LAMA2 related dystrophy or collagen 6 related dystrophy between ages 5 and 16. years. And the outcome of that phase 1 study was the compound Omegapill was found to be safe and with a very favorable pharmacokinetic profile. The sponsor at the time, Santhera, had prepared plans for a phase 2 study, which they presented to the Treat NMD Advisory Committee for Therapeutics, or the TAC committee. And the feedback from the TAC committee was that although there was some very nice preclinical mouse data of the use of Omegapill in 2 mouse models for LAMA2, so the DYW and the DY2J mouse model, the TAC committee said they felt that there would need to be further work both on biomarkers, but as well as further preclinical data for patients with the collagen 6-way dystrophy subtype if it would go forward to phase 2. And so we've been working with Santhera and other collaborators in the field to see how we could kind of push forward the work towards identifying biomarkers as well as getting more preclinical data for the collagen 6 patients. So the current time, there's not a clear pathway forward for moving on to phase 2. If there would be identification of biomarker, it probably would fees that transition. So that's kind of the current state of things with Omegapill in particular.
So not dead, just on hold.
Course, because we have also been having conversations with companies and academics who are working on other therapeutic approaches with the idea that we could use Omegapill as an adjunctive therapy with those other therapies. Omegapill really works more on a downstream type of pathway in both LAMA2 and collagen 6-way dystrophy. So ultimately, probably would be effective as an adjunctive. So it's definitely not dead, just kind of trying to figure out the best pathway forward.
Can you quickly define what downstream means and how that differs from like gene therapy?
Sure. So what Dr. Bönnemann presented this morning with the therapeutic toolbox, so often we try to kind of go to the level of the transcript, modify or affect, silencing of one allele. You're trying to get to the kind of the heart of or the core of the matter to correct the transcript. With Omegapill, it's not specific to the genetic abnormality. It's really a secondary effect of the mutations in the LAMA2 gene or the collagen 6 genes, which cause this on a cellular level. And so it's not going to the core of the genetic underlying etiology.
Great, thank you. We have a question. Have there been any research on carriers with manifesting symptoms for LAMA2? Maybe we want to explain first what manifesting carriers are, and then whether or not this is an area of interest in research. I'll leave that to anyone. Dr. Bönnemann or Dr. Muntoni, would you like to answer those?
So the answer is this is a recessive disease. It's truly a recessive disease. That's why I pointed it out on the slide, and that's different from other disorders that we discussed this morning. What recessive means that if an affected child needs 2 affected copies, one from each parent to have the disease manifestation. However, each of the parents who's carrying only 1 copy on the background that is mutated on the background of a normal copy is unaffected. And that's really what recessive means. Recessive means it is hidden by the normal copy unless you have 2 affected copies in an offspring. So whatever is manifesting, I suspect there is a parent who is manifesting neuromuscular symptoms. That has to be something else. It's not the carrier status of merism.
Maybe just to add, the good news about what Carsten mentioned is that as for therapeutic application, you will not necessarily need 100% of what is deficient, because the carriers, as Carsten said, they are invariably normal. And therefore 50% will be enough to be normal. And then presumably even less will be enough to have a significant clinical benefit.
Great. Okay. Thank you. Another question. In 2018, Dr. Newhouse said it will be important now to package the linker proteins in 2 different AAV vectors and deliver them at different times after birth to see if it still works to protect the muscle, as this disease is already established at birth. Well, realistically before birth, right? Then we can think about a clinical trial. Do we have any updates on this topic?
It goes to Markus.
Yeah, okay. But of course, this is on mouse work, but we and mice develop these symptoms quite early. And we have so far treated mice with AAVs at the age of 1 day, which is an early time point. we see benefits. We have also tried to use, or we injected into 3-week-old mice, and we see some benefit, although this is of course much smaller. And the transgenic mice I presented also allow us to induce the expression of those linkers later in life to test exactly this question. And it looks as if we can get improvement, but of course this is just less as when you do it at the early stage. But we have the tools to test this, at least in mice. And I should also emphasize, mice develop really fast. I mean, they gain weight from 1 week to 4 weeks. It's a huge increase in weight from 1 gram to almost 20 grams. And this is, of course, never reflected in a young child. I mean, the development there is much slower than in mice. This is, of course, the challenge in this particular mouse model, because it really reflects the human disease quite well. It's a severe form of a muscular dystrophy. We don't have much time to treat those mice.
Great. Okay. Thank you. So we have about 4 minutes left. I'd like to go to each of you and kind of give a wrap-up statement, kind of a forward-looking or a statement of hope for the patient community. Dr. Reghan, Dr. Foley, we'll start with you.
Sure, that's fine. So I guess kind of, coming from the perspective of the clinical research team that's been preparing patients for clinical trials, it's very exciting to see that very likely, we're realistically optimistic that there'll be multiple options for our patients. And so our job now is to gather some very early natural history data in patients 5 years of age and younger, so that we are ready when these therapies are going to be translated to the patients.
Great. Dwi, would you like to give a statement of forward-looking statement?
I echoed what everybody was saying earlier during the talk. So with from Marcus, I think Marcus pointed out that we need a company support to actually do that. And I think as much as we can do all the science very well, and the NIH is wonderful at giving funding if we deserve that. But I think to move this to really therapeutic opportunity, we do need company support.
Great. Veronica?
Yeah, I think research is moving in tiny little steps, but every step is still something forward that it can put us forward into the development of a therapeutic approach. So it's important that we don't lose hope, despite acknowledging that this development can take time.
Great, thank you. Professor Rüegg?
I'm of course biased, but my dream would be, because at my age, my dream would be to see linkers a patient being treated with these AAV linkers to me. And I'm still hopeful that we will get there sometime soon, hopefully.
Wonderful. Dr. Bönnemann? You're muted.
I have to say, I'm really excited by the development of what we've heard today. You know, we start out with an unwieldy, huge molecule, and where we said, you know, we'll never get to a therapy for this one. That's too big and too complicated to do. And now we have two highly ingenious approaches, a linker approach that's very feasible, and upregulation of a rescue gene that's very feasible to do. This is remarkable, if you think about it, to be able to tackle this disease in this way. But still, it's a mountain that is in front of us. I mean, this is a disease that's fully expressed at birth. So we really have to be as efficient as we can probably be to turn this around to make it better. And hence, I think there is room for research on all fronts for this disease. In the end, we'll be probably looking at combinatorial treatments where we address the root cause and then make the muscle more resistant, more effective at regeneration. This is not one answer fits all. This is an effort that the entire research community has to be behind.
Absolutely. Professor Muntoni, you want to wrap us up?
I just think that the technology advancement that we heard today, they are just very, very impressive. And so congratulations to their colleagues. I think that a lot of information is being gathered in terms of how many of these vectors and so on, and work in other conditions. So the field is not only the lamin alpha 2, but is also the entire neuromuscular, and there will be a lot of cross-learning. So I think this is encouraging. There will be still a little of time, but I think I'm sure that Markus, before he retires, will see this, and we want to see that with him.
Absolutely. We have a lot of activity going on in LAMA2 and lots of hopeful treatments coming. So I thank you so much for your time today. Fantastic panel. If you have additional questions for our experts, please email us at scifam@curecmd.org, and we'll do our best to get some answers for you. Thanks so much, everyone.
Thank you.
Thank you.